Hi, I'm Caroline Clark, conference producer at Technology Networks, and I'm here to introduce our next talk, Disrupting Crop Breeding with Genome Editing and Haploid Induction. I'm pleased to welcome Tim Kelliher as your presenter. Tim supports a program of new breeding technologies from research through implementation that accelerates Syngenta's breeding and trait delivery pipelines. Tim received his PhD from Virginia Woolbott's laboratory at Stanford University, and his undergraduate work was completed at Swarthmore College. A warm welcome to you, Tim. Following the presentation, we'll have a Q&A session, and we'll welcome any questions that you may have. Please ask questions using the Q&A system to the right-hand side of the video player. You can also use this feature to connect with attendees and discuss the talk, so feel free to take advantage of the chat system by introducing yourself and sharing your thoughts during Tim's presentation. If you experience any technical difficulties during the presentation, please request help using the technical help icon on the left-hand side of the player. Now, without further ado, I'll hand over Tim to begin his presentation.
Yeah, thanks for joining today. I'm really excited to talk about some work that we've been able to publish in the past 10 years here at Syngenta. A lot of this work was accomplished with the support of many team members. You know, Syngenta really has a team-based operational strategy, I guess you would say. A lot of the projects are done with people from all levels of research, people with PhDs and master's and bachelor's degrees, all working together to deliver innovation. And I got some great examples of that today. You know, I work at this site, Research Triangle Park in North Carolina, which is one of our two biotech centers in the Seeds organization. The other biotech centers in Beijing, and we also collaborate with that group quite a bit. I'll just jump right into the story here. You know, Syngenta has been working on genome editing in several different crops over the past, 12 years actually, back when meganucleases first started to become something you could apply in crops. And then, of course, with CRISPR coming online, we started to increase our efforts in genome editing. And this story, you know, really intricately involves the development of genome editing technology as well as the sort of discovery and implementation, I guess, of how we can use genome editing with other advanced breeding technologies, such as double haploids. So I'll start with this, again, introduction of working on how genome editing and haploid induction have been able to kind of work together in our labs to bring new innovation across different crops. We'll start with why is double haploid breeding important? In corn, actually, double haploids are one of the critical success stories that made corn continue to increase its genetic gain and be profitable for farmers to grow. And that's because double haploids is something that allows our breeders to accelerate the cycle time of the breeding process. And this because basically it allows the production of inbred lines that you can have fixed novel genetics in for testing traits in the field. It allows us to produce those inbred lines, those critical inbred lines for testing in just 2 years versus the traditional 7-year process required for purity, genetic purity. The way that this works, I'll share on the next slide, but I'll start with this maize story because while all crops want double haploid breeding, maize is the only one that we've really been able to deploy it at a massive scale. And I'm not just talking about Syngenta here, we're talking about all the major biotech crop breeding companies. And so we'll start with the basics of haploid induction. What we have here on the top and then on the bottom are two different forms of haploid induction, one in which the inducer is the female on the top, and the other one is where the male is the haploid inducer. So in haploid induction, we have a pollination between two lines. There is an attempt at normal fertilization where the haploid genomes of the two lines come together in the zygote cell to fuse, but then something goes wrong in that fusion and initiation of early embryo development. And in the case on the top, The reason something goes wrong is called SNH3. I'm going to just put a pin in that one, and we'll talk about SNH3 later in the talk today. And the one on the bottom, which is the one we'll start with, the corn system, the thing that goes wrong is based on the MATL gene. And again, that's in the male. So during haploid induction, a lot of the times normal fertilization haploids to make a typical F1 diploid hybrid. But then some of the embryos will experience this genome loss after fertilization, resulting in only the chromosomes from the wild type parent being maintained in the progeny. And that's a haploid progeny, so it only has one set of chromosomes from the wild type parent. And so we'll start with the maize system on the bottom. So again, double haploid technology is now like the foundation of maize breeding. People basically, all the breeders, when they make new starts, they take two parents that have different interesting traits that they want to combine. They go make an F1, and then they go right into the haploid induction and DH process. I've got some nice videos here on the left of haploid nucleus versus a diploid nucleus. You could see how the cells are smaller. The nucleus has half its number of chromosomes in the haploid, and the plants end up smaller as well, and they're sterile. So in the DH process, you use the haploid inducer as a male, pollinated onto any F1 or F2 new breeding material. Genome loss happens during fertilization. You get the haploid embryo, which you identify by virtue of a color marker. So this is a natural anthocyanin-based color marker that's in the haploid inducer line. Some of these lines do have purple leaves, even in purple anthers and purple tassels. And the diploid offspring will have a purple scutellum in the embryo, and that is because the male genome is present. Those are the normal diploid hybrids. Then the haploids here are in yellow. Those are the ones that lack the male genome, and they only have the chromosomes from the new breeding start, the F1 or the F2, that has now been recombined in the ear. So each of these haploid embryos will be genetically different from each other. And you want to then identify those by that color marker, take them out, double them, which you can use using colchicine, which is a mitotic inhibitor. It interferes with the assembly of the microtubules in the spindle, so you can't actually go through a normal cell division, and this allows for a doubling of the total number of chromosomes in a cell, basically producing an identical copy of every chromosome. This was kind of shown on the prior slide. If we go back here, this slide, where you can see that mitotic inhibition process, basically a copying of every chromosome. This allows a haploid to become a doubled haploid, instant inbred line, so that whatever that recombinant state is after the recombination in the ear on the females, you then produce an instant inbred line of those crossovers on all the chromosomes. And then each of those individualized DH lines are then analyzed, either using genetic markers, if you know which traits you want to select for, or in the field through phenotypic testing. And then those can then be recycled at the beginning of the cycle by making new parent crosses, or they can just be moved on. If they're really, really valuable, they can be moved on in the breeding cycle forward. So that's the basics of DH breeding and how it kind of works in a DH lab. I'll now move on to what Syngenta specifically did in this area for maize, which is we wanted to really understand why the male inducer in maize works so well, because again, there's a lot of other crops that wanted DH technology, but they didn't have it. And so we figured if we could identify the gene behind haploid induction in maize, we might be able to develop it in different crops. So Brent and Satya are breeders in our seeds development organization that began working on developing the haploid inducer lines at Syngenta, and then also started a mapping project to try to identify the genetic trigger behind the trait. And they were able to map the trait to a small interval on chromosome 1, which then got narrowed even further, and it basically down till it was just about 7 genes. And at that point, we wanted to know which of those genes had mutations or differences between normal corn and haploid inducer corn. And so we found 2 by sequencing those genes. There were 2 genes in particular that had major disruptions. One was missing in the inducer, and the other one had a 4-base pair insertion, which caused a frame shift in the amino acid chain. I'm just showing the C terminus of this gene, and you can see a little frame shift producing novel peptide there, and then a stop codon. So in order to figure out which of those two changes were responsible for haploid induction, we needed to be able to transform the wild type copies of those genes into a haploid inducer line. Challenge there, though, is that most haploid inducer lines are not transformable. If you know much about maize transformation, you probably know that it's a huge challenge, and that most lines are not transformable. And so being the prescient thinker that he is, Brent Delzer, the breeder who had mapped the QTL, had actually started developing a transformable haploid inducer line way back in 2007. So that by the time I came on board Syngenta in 2013, he said, not only have we mapped the gene really close, but I've developed a line of corn that has this QTL in it, but it's now in a transformable background, a line that's known to be transformable. So he introgressed that QTL in to that background. And that was supremely useful because it allowed us to put wild type copies of the two candidate genes into the inducer line through standard agrobacterium-mediated transformation. And then we were able to test which one fixed the haploid induction trait. Now, for this purpose, there's several ways that we measured haploid induction trait in general with the purple color marker and genetic markers and everything else. For the purposes of this slide, I've just simplified the whole thing and shown the trait as what it looks like when you pollinate a random line of corn with haploid inducer pollen. This is actually what you get after about 15 days. So you get this really spotty ear and some no kernels, some little white kernels that have the endosperm report, and then some normal kernels. It's actually inside those normal looking kernels that we'll find that normally the little haploid embryos. Those haploid embryos are not smaller. They're not there's no other way to distinguish them from the normal diploids. About 85% of these yellow kernels will have diploid embryos in them, depending on how good your inducer is. And about 15% will be haploids. So this looks like an ugly ear to most people. I realize that, of course, again, the whole point of DH is not that you're trying to breed this. This is a means to an end. It's a means to making those DH lines that are then so useful for breeding because they take the breeding bottleneck, the breeding pipeline process, which normally takes 7 years per cycle of making inbreds, and scrunches it down into 2 years or 2 generations. So it's a huge time saver. So I know that ear looks ugly, but that is what we want in a DH pipeline. And anyway, so after we had transformed this line with the 2 candidate genes, one of them didn't change the phenotype. We outcrossed the transformed events, and the ears look just like that. But then after we transformed the normal copy of the matrilineal gene in, it did change the way the ears looked. And so this is the PGM example. And then with matrilineal, we got a normal pollination. And we got no haploids out of, or like a 0.03% haploid rate or something very low out of that when we inserted the wild type copy. And then this is around the time that CRISPR came, and so we did a TALEN-based gene editing, which we had been working on for a few years, along with meganucleases. But then when CRISPR came along, we repeated the experiments with CRISPR, and we found that if we knocked out the wild type copy of the gene in a normal line of corn, we could make a novel haploid inducer with about a 5% frequency of haploid induction. So that wraps up the maize cloning of the matrilineal gene. And I do want to say that there is another important gene in maize that is responsible for making your haploid inducers more efficient. So the matrilineal knockout alone will only give you about 5% induction rate. And in some lines, it's been even lower than that, like 2%. But if you combine that with another mutation on chromosome 9, you can enhance the rate of haploid induction. And I'll show that on the next slide. Before we go there, I just want to share really quickly what the identity of the protein product for matrilineal is. So it's a phospholipase, and it localizes to sperm cell membranes. Now, we originally showed this in our 2017 publication, and you can see the little movie on the right side of a pollen grain with 2 cells lit up in the middle. Those are actually the sperm cells of the pollen grains. There's 2 sperm cells in every pollen grain. In fact, if there's one thing I want you to learn from this talk, it's that you remember that there's 2 sperm cells in every plant pollen grain. Most of you probably already know that, but for those who don't, it's a great little trivia fact. If you ever get a quiz on plant reproductive biology, it doesn't come up in trivia very much, but it's good to know anyways. So yeah, those are the 2 sperm cells. You can see they're very wispy. They kind of look like cat eyes, or like somebody put on way too much eye makeup. They've got little nucleus in the center. And then those wispy things on the ends are the really long extended membranes, which form the male germ unit. You can see in a germinated pollen tube that those membranes are really long and stringy, very strange looking things that kind of get dragged along with the vegetative nucleus as the pollen tube grows down the style. And in corn, of course, you may know that those silks that you have to pull off when you eat your corn, those are very long, basically tubes that the pollen grains have to grow down to deliver the sperm cells and their membranes to the embryo sac. And we also found the localization of the matrilineal protein, not only here on the outer membranes of the sperm cells in the pollen tube, but even inside the actual embryo sac after fertilization. Now, another group out of France has done much better work than we did on this, much more extensive characterization of the membranes and the localization, and I encourage you to look further into their work. They called matrilineal not like dad, which is a cute and clever name because the dad's genome goes away, and they proved that the localization is actually on the is coming from the vegetative side and kind of adhering to the membrane that surrounds the sperm cells. So if you take a sperm cell promoter and drive the matrilineal GFP fusion with a nuclear localization tag, it doesn't show up in the sperm cells. It's actually a vegetative derived protein that kind of coats the sperm cell. So that's interesting. We haven't really been able to figure out the core mechanism behind any of this, about what is actually happening with the membranes, but there is one fascinating publication out of China, I think CAU, Chinese Ag University, that showed that the sperm in the haploid inducer, the sperm membrane, sperm chromosomes actually fragment, undergo kind of a self-destruction process, even in the pollen before the fertilization occurs, which may be why the haploid induction happens. It's a solid hypothesis there. So moving forward, getting away from the mechanism and actually what was our original purpose at Syngenta, which was to learn the mutation so we can transfer it to other crops. We looked at the phylogenetic relationships as well as the synteny, and identified a matrilineal ortholog in rice. And then we're able to show that it also is expressed in pollen grains. And by knocking out that gene using CRISPR-Cas technology, our Beijing group, our innovation team over in Beijing, was able to show that you could create a haploid inducer line in rice. This is our first haploid inducer line in rice that's been published. I think to date, there may be a couple of other cases coming out soon, though, of other genes that work as well. So outside of the matrilineal gene, I did promise that there'd be an enhancer gene I wanted to mention, which is DMP. So this is a gene on chromosome 9 in maize, which enhances the haploid induction rate of matrilineal. And if you independently knock out this gene in diverse crops, you can get a low haploid induction rate of like somewhere around 1% or a little less. So matrilineal, it turns out, is really grass specific. And so finding the homologs of matrilineal in dicots like tomato or soybean, for instance, it's really hard to figure out what's the actual gene to modify there. But for DMP, it's an enhancer of haploid induction in maize, and it's its own haploid induction system on its own. If you just knock out DMP in diverse monocot and dicot species, you can get haploid induction. And on the right here, I just want to share our data from maize showing that if you lack the QTL behind DMP, which is called QHIR8 in maize, you have a lower induction rate, and if you have it, it boosts it higher. So again, DMP, the QHIR8 is the QTL that has DMP in it. I think there should be some more work coming out on understanding the mode of action of DMP and as it relates to matrilineal in the next few years. All right, if I haven't lost you so far, I'm going to pause the story there and switch to a totally different haploid inducer system, which I referenced earlier, which is the SENH3 system. Now, this one is pretty fascinating because instead of being used as a male, you use it as a female. So in this case, the female is the inducer, and the male is the breeding line that you want to make haploids from. So a bit different from many perspectives. First of all, it's a different genetic mode of action, and the consequences of the haploid induction are very different. And that's because if we kind of back up a little bit, fertilization between female and male in plants, those just like in animals, the cell sizes are very different, and that has very big implications. So one of the biggest implications of it is that the cytoplasm of the egg cell is what is inherited by the offspring. And in the cytoplasm, of course, you have the mitochondria and chloroplast, which have their own genomes and genetics. Those are maternally inherited. And so, cytoplasm genetics does play an important role in crop breeding, but it usually is ignored and not very well studied. People always are mapping genes and finding genetic relationships when you talk about the chromosomes in the nucleus, but the cytotype is often ignored or not studied. So when you have a SENH3 haploid inducer, you have the female having the inducer is on the female. And so this, by the way, this sort of nucleus here is very different as well. It has very open chromatin state. There's a lot of gene expression coming from these chromosomes, whereas from the male, it's a very compact state. And so the early days of the embryo development are very much dictated by what's happening in the female side. This nucleus will get transferred and fused in a normal situation. But in the haploid inducer situation, this male nucleus will come in and this female genome will eliminate. And so now you've basically put the male nucleus inside the female cytoplasm. And so those cytoplasm genetics are going to be inherited into the haploid. So that means that this female inducer, based on the SENH3 gene, makes haploids that are cytoswapped. And this brings in several applications. So not just DH breeding, but the ability to directly transfer cytotypes into different nuclear genetics so that we can begin to start understanding the effect of the cytoplasm on the phenotypes of the crops. So it's kind of a whole new genetic angle here. I would also share that for the sake of hybrid production technology, CMS, cytoplasmic male sterile lines, have a very particular cytotype. There's something wrong with their mitochondria that causes male sterility. Those lines are extensively used in wheat and rice and corn, in sunflower and tomato, and all of our, many of our hybrid crops, they use CMS. And those are normally delivered through an introgression process, which takes several years. This cytoswapping process can replace that. And so cytoswapping not only has the chance to enable novel genetic discovery, but it has a chance to simplify our hybrid breeding production systems. Okay, so what I want to share now is a story of how we were able to develop a novel SENH3 inducer in wheat. So the SENH3 story actually starts out at UC Davis about 10 years ago with the publication by Ravi and Simon Chan on the modification of the SENH3 gene in Arabidopsis. It was a very exciting publication at the time because, of course, people realized, hey, we have a female haploid inducer. It can cytoswap. It could do haploid induction in any crop. Now, SENH3 stands for centromeric histone 3. It's a very conserved histone protein that forms that's responsible for recruiting the kinetochore proteins to the centromere and is responsible, therefore, for normal cell division. And so when they realized that they could create haploid inducers through modification of this essential cell division protein, and that protein is present in all crops, it seems like a great opportunity for developing haploid inducers across crops. However, after 2010, there were years and years of research which were just marked by frustrated graduate students and money that was spent that did not yield much in these diverse crops. SENH3 proved itself to be a very challenging gene to modify in diverse crops, probably because it's required for every cell division. You know, there's 2 domains in SENH3, and what Ravi and Chan had done back in 2010 was modify the N-terminal domain, which is what we call the tail. This is the thing that sticks out from the histone core particle and is responsible for recruiting kinetochore machinery. The modification that they did not disrupt the even more conserved, hyper-conserved histone fold domain, which actually binds the core DNA particle in the histone in the nucleosome. So what we did in wheat was kind of learning from what had failed in other crops. We tried to develop a direct genome editing strategy where we could either cut out a whole segment of the tail by having 2 guide RNA cut sites that then allowed that segment in between to drop out and be deleted, or those same guide RNAs, if they didn't cause a dropout, could, in theory, if we designed them well, cause the, and this is the start of this is the wild type gene in green, could cause a frame shift that would alter the amino acid code for the intervening sequence between the 2 guide RNAs, and then a second frame shift that was complementary to the initial could restore the normal frame. So this, we called it the restored frame shift strategy. And what it would do is create this altered N-terminal sequence, which may be less important for normal plant growth and development, but without altering the histone fold domain, which is basically essential. And we thought that this might lead to a haploid inducer, whereas other approaches in corn, such as GM complementation strategies and other approaches, didn't work. Not in corn, in diverse crops. Sorry. So this restored frame shift or large deletion approach yielded a diversity of alleles in wheat. And in particular in wheat, we focused on the A genome gene target of SENH3. There are 3 copies of this A, B, and D genome, as well as the alpha and beta versions of SENH3. So our editing created a whole diversity of mutations, and we were basically looking for the knockout of the B and D alpha versions and the restored frame shift of the A alpha. And we were able to, after a lot of work, combine those mutations. And I encourage you to check out our publication on this from 2020, where we actually were able to generate about 7% haploid induction rate in wheat using the restored frame shift strategy. We did that with 2 different types of heteroallelic combinations. In one, the restored frame shift was made homozygous, and in the other, it was heterozygous over a wild type copy of the A allele. The interesting thing here was that actually the heterozygous version, where we left 1 copy wild type, was able to produce the most haploids, about the 7% rate, whereas this one where it was homozygous was only able to make about 1% haploids. So I think the lesson from this is that, first of all, SENH3 is way more complicated than we thought back in 2010. But second of all, that there are creative ways to go about trying to create these haploid inducers, and heterozygous states may be the right things to think about. The reason that may be is that as the cell goes through meiosis and prepares for female gametophyte development, I think we want the state of those centromeres to change at that point. So if you're in a heterozygous state, and then the wild type copy goes to one gametophyte and the restored frameshift goes to the other, then suddenly that one that got the restored frameshift no longer has that wild type copy around anymore after meiosis. And so its centromeres then may change more quickly than the other one, which creates a possibility to get the haploid induction. I think the theory behind this is still being developed, so take that with a huge grain of salt. And let's say that the story is definitely not over for SENH3, and I think there should be more progress in the future. One really exciting development that actually confirmed the sort of thinking around the heterozygous being important is that in maize, Kelly Dawes' lab at Georgia actually showed that an extremely simple knockout of just one of the copies, leaving a SENH3 wild type over knockout combination, can give a 5% or 6% haploid induction rate in corn. This is an extremely simple mutation to create, obviously, using genome editing. Basically, any knockout will do when it's over a wild type copy. So the turning now away from haploid induction in general and thinking about what else can we do with haploid inducers to enable our genome editing ambitions. So I want to start with the idea that I said back at the beginning, which is that genome editing platforms are being built in multiple crops, and they're capable of making all kinds of different precise edits and creating lots of different traits. I think one of the critical challenges here to genome editing being deployed more globally is that there is going to be a bottleneck of delivering those traits to haploid inducer lines, or excuse me, to our commercial portfolio lines. And that is because, again, it is really not easy to transform just any line of a crop. And, a breeding company like Syngenta, has dozens of market segments, and each market segment has, many, many different specifications for different types of materials that need to be developed. So there's literally hundreds of lines, thousands of lines in development at any one time. And if you want to bring edits to all of those lines to deliver a new trait, it creates a huge breeding bottleneck. And we can't just directly transform them for cost and scale reasons. So the general process that would be used to find and deliver a new novel edit would be to transform one line, evaluate how it looks in the greenhouse, and then introgress the edit into representative elite lines, evaluate their genetics by gene, by environment performance and field testing, and then if that works, introgress it into all of the elite commercial portfolio and deliver it to the market. This just generally takes too long, and especially for non-GM traits, the value may not be there. And so what Chideng is the head of transformation. Actually, he's now the head of the pipeline delivery here in our Research Triangle Park facility. And he was thinking about this problem one day, and I was telling him about the matrilineal story and how we hadn't really been able to solve the mechanism of haploid induction in corn, the sort of detailed mechanism. And he said, well, he's been looking for a way to edit commercial lines directly. What if we put a CRISPR construct into a haploid inducer line and then outcross it? Will that CRISPR be able to fertilize and edit the haploids? And this simultaneously, this idea would have the potential to solve his bottleneck problem and answer the question of the mechanism of haploid induction. And let me explain what I said right there. So in haploid induction in maize, again, we were trying to debate between the two concepts of, do haploids come from a failed fertilization attempt where the DNA is from the male is actually never delivered, or does it come from a normal fertilization followed by loss of the male genomes from like sort of expelling the male chromosomes from the nucleus of the hybrid? And so this kind of pre-fertilization, post-fertilization duality was the subject of our thinking at the time. And Chideng thought that, well, if we just load the inducer up with a CRISPR construct, then if we find edits in the haploids, then there was some kind of nuclear fusion and fertilization that occurred to give rise to those haploids, because that CRISPR had to be delivered to make those edits. So this idea became, I think you can probably see it more easily in this graphic, where the haploid inducer pollen actually carry a CRISPR transgene, and then fertilization of the sperm in the egg with the sperm enlarged for visual effects leads to a transient zygote state where editing can occur on the female chromosome, and then the male genome is eliminated, and then genome doubling occurs, and that star represents the new edit and the edited doubled haploid line. So if this worked, we could basically use a CRISPR positive haploid inducer to deliver editing traits directly to our commercial portfolio, with skipping the time-consuming backcrossing introgression process. And this worked the first time we tried it in corn, and the efficiency was about 4 to 5% of our haploids were edited, and it led to the patent protection of a concept called high edit for haploid induction genome editing. So again, high edit allows us to skip the several generations of backcrossing involved in normal trait introgression, allowing us to more quickly bring genome edits to our commercial portfolio of lines in maize. After working on this, we were able to show a yield improvement traits, both in kernel number and kernel weight. And then we extended this high edit concept to wheat using a Y-cross system, where we actually pollinated wheat with corn pollen, which is a common way to make wheat haploids, and then found edited wheat haploid plants after rescue. So this way, the corn pollen actually delivered the CRISPR to the wheat. So that's pretty neat. And here's some of the genetic data from that experiment. And then finally, building upon the SENH3 work, we were able to show high edit works really efficiently in Arabidopsis and the SENH3 haploid induction system. In this case, the wild type was a Landsberg erecta, and the inducer was a Columbia with the SENH3 haploid induction trait, and then we used Cas targeted to the glabrous1 gene and showed that about 17% of the haploids were edited. And so this is much higher efficiency than maize, about 4-fold higher. And that may be due to the fact that if we go back to my slide of the egg and sperm cell, the female genome with that open chromatin may be able to drive a higher expression of CRISPR during the fertilization process, although there's several other reasons, such as genome size, that may be responsible for that higher efficiency of haploid editing. So this is the last slide before the end. I just want to say that with high edit, Syngenta, is now working on deploying edits to any variety of corn, and we're working on the technology in other crops. And I want to share that, if researchers are interested in using high edit technology, please reach out to me or to our technology integration group at Syngenta. It opens up the door for a lot of interesting, answering a lot of questions in both research and product development. And we've used CRISPR to knock out genes, this kind of high efficiency, just cut a gene. But we are, of course, exploring ways to use the whole full Swiss Army knife of CRISPR technology to base edit or silence gene, amplify expression of the genes, or replace gene sequences to create new alleles through high edit. And so with that, I will just share this acknowledgement slide and take any questions.
That's fantastic. Thank you, Tim. That was a fascinating talk, and I'm sure we're going to have lots of questions. And from our audience, and I can see that we've already received some. So I'll start with the Q&A now. So the first question we've had in is Baby Boom or Wushell technology from Corteva an alternative to high edit, HI edit?
Yeah, that's a great question. Yeah, so I mean, so Baby Boom and Wushell, for those of you not as familiar, is a way to transform elite lines of different crops. It works pretty well in corn. And so theoretically, you could, the sort of transformation bottleneck I was speaking to, you could overcome a lot of those challenges with that technology. I think the difference is that high edit would allow you to really efficiently deliver edits to a commercial portfolio without actually having to transform different lines. And transforming lines is still an expensive process because you use the greenhouse step, you have to do the lab step, several months of tissue culture. So I'm not sure that Corteva or other groups like them are using the baby boom Wushel system to deliver edits to a full commercial portfolio, because that would be asking too much. So I think high edit does have there is some overlap there, but high edit should be a more efficient system in general for the commercial portfolio delivery.
That's great, thank you. I'll move on to your second question. What's the regulatory view of high edit compared to other methods?
Right. So it's definitely an unresolved question, and one I'm not probably I don't have enough knowledge about to speak to. Um, I It really depends on which geography you're talking about and how the genome editing as a biotechnology, is regulated specifically. In some jurisdictions, the way that the edit is made might matter, and the fact that there's no CRISPR transgene ever inserted into the commercial line, the line that we make the haploids from, might make a difference in how those edits are sort of evaluated and regulated. But I think it's too early to say if there for sure is going to be any difference between the way a high edit is regulated and one that's sort of delivered through standard transformation.
That's great, thanks, Tim. And your next question is, do we know why haploids are induced in the corn system?
Right. So yeah, I think I tried to answer that, but it's I guess we don't. There's. I would just say, like, if you don't go looking for publications from me on that anymore, I don't think. Or maybe some people in my group might have some informative publications at some point here, but we have kind of stopped exploring the real fundamentals of the way that phospholipase might work. And there's several other groups that have followed up. Again, the group out of France calling the gene not like dad. Those have done a lot of great work on getting towards the mechanism, and it may be this chromosome fragmentation thing that's been hypothesized out of China, out of CAU, but I still think we don't really know. One of the problems is really getting inside of a corn kernel to sort of, if we had the perfect microscope that could really see exactly what's happening at fertilization, that would be wonderful, but it's pretty hard to get in there. So I still think that we've got some work to do to figure out the real mechanism.
That's great, thank you. We've got time for a couple of more questions. And your next one is, how easy is it to transform the haploid inducer line in corn and other systems?
Yeah, so we have so the original line that Brent Delzer developed had like a 3 to 5% transformation efficiency, which is okay. And we certainly have worked on improving that. There does seem to be like a kind of basic challenge because haploid inducers, as you saw with the ear photo, they don't make a normal ear, which you kind of need all those embryos to be developing normally to have an efficient transformation system, as well as the genetics. So when you self-pollinate a haploid inducer, it doesn't make like a nice batch of embryos to transform. So I think there's some fundamental incompatibilities there that are that will cause struggles. So I'd say that's kind of an open area for improvement. We have made some strides internally that we haven't been able to publish yet, but yeah, it is certainly one of the challenges for high edit. But I do think that that baby boom bushel technology can also help alleviate some of the transformation inefficiencies to enhance the rate there.
Thanks for that. And your final question, it is, what is the situation with SEN-H3 haploid induction in different crops? And also, what about doing microspore editing for elite line edited DH?
Yeah, so there's been a lot of, as I mentioned, the progress in corn. It's really exciting in SEN-H3. I still think, I know there's been a ton of work in tomato in SEN-H3, and it seems like almost anything folks try doesn't work. Similar for rice. It's just people have thrown, the kitchen sink, so to speak, at the problem and not been able to get it to work. I do wonder, I don't know, but I do wonder if some crops might um, the need for SEN-H3 in normal development is more than what we can do to manipulate the sequence to get a haploid induction trait. So like it's almost like a trade-off. Like if you compromise it enough, you could get haploids, but for some crops, you're not able to sustain normal development with that. I think that might be what's happening there. But it's still certainly something that needs to be explored more. And in particular, if people can make minor mutations in SEN-H3 and maybe stack those with modifications to other kinetic core proteins, that could be an approach. And then the microspore editing. So again, I think the challenge with microspore editing as a sort of people think of it as like a holy grail solution for this. It could be in some ways. You'd have to kind of always regenerate those microspores, which is not easy. Depends on the crop. Efficient delivery to microspores is really challenging as well. So it is a good way to make haploids in DH lines. Getting editing to occur during that process and regenerating those edited lines, you need a lot of work there. And I think it could be something that we could see happen in some small crops, but maybe not the near future for major crops because of some of the challenges associated with microspores.
That's great. Thank you very much, Tim. And unfortunately, that's all we have time for today. Any questions that we didn't have time to get to will be sent to the speaker after the event. And as with all the other talks in the event, this talk will now be made available to view whenever you want for the next 30 days. That just leaves me to say thank you, Tim, and we will be back soon with the next talk.
Thank you.